Hello and welcome everybody to my channel Lumini Bioentrance, and today we will be discussing GATE Life Science question paper of the year 2024. I will be discussing the biochemistry section. If you are new to my channel, please do like, share and subscribe. So coming to the first question. Which one of the following pairs of amino acid is not incorporated in a polypeptide chain? So 4-hydroxyproline gamma carboxyglutamate. They are post-translationally modified amino acids and they can be found in proteins. 4-hydroxyproline, it is found in collagen. Gamma carboxyglutamate, it is found in proteins as well. So the next one, gamma carboxyglutamate and desmosine. They are all, desmosine is also post-translationally modified and it is found in elastine. Then ornithine and citrulline, they are not found in any of the proteins. They are intermediate in the urea cycle. Then 4-hydroxyproline, 4-hydroxylysine, they are found in collagen. So C is the answer. Then mammalian cells cultured at low temperature lead to an increased sterol content in the membrane. So elevated sterols in the membrane result in. So mammalian cells, they are cultured at low temperature means they have the tendency to freeze. And this can hinder with the transport and other cellular functions. So to counteract this, the cell has increased the sterol content of the membrane. So because of this sterol content, there will be enhancement in membrane fluidity. So answer is option A. Then next one. Which one of the following metabolic intermediates is common in glycolysis, nucleotide synthesis and glycogen synthesis? So it is glucose 6-phosphate which is common. In glycolysis, glucose 6-phosphate is converted to fructose 6-phosphate and then finally fructose 6-phosphate to fructose 1,6-biphosphate and finally to pyruvate. Then in nucleotide synthesis, glucose 6-phosphate is converted to ribose 5-phosphate which is involved in nucleotide biosynthesis. Then in glycogen synthesis, glucose 6-phosphate is converted to glucose 1-phosphate and is used in synthesis of glycogen. Citrate is an oxaloacetate intermediate of citric acid cycle, and glycerol 3-phosphate doesn't have anything to do with glycolysis, nucleotide synthesis, glycogen synthesis. It is involved in lipid metabolism. Then next one. In mammals, hematopoietic stem cells that give rise to different type of blood cells are known as. So hematopoietic stem cells, they give rise to different type of blood cells, that is myeloid cells and the lymphoid cells. The myeloid cells, they give rise to red blood cells, platelets, monocytes, and lymphoid cells, they give rise to T cells, B cells, natural killer cells, etc. And hematopoietic stem cells, they are considered as pluripotent because they have the ability to differentiate into multiple cell types. Now totipotent stem cells. Totipotent means they have the ability to differentiate into all types of cells. But hematopoietic stem cells don't differentiate into all type of cells. They differentiate into multiple cell types. Myeloid cells and lymphoid cells, and from myeloid cells you will get the other red blood cells, platelets, monocytes and lymphocytes. Lymphoid cells you will get T cells, B cells, etc. They are pluripotent stem cells. That is correct. B is correct. Myeloid progenitor cells and lymphoid progenitor cells are not arise from the hematopoietic stem cells, but they are not stem cells themselves. So the answer here is option B. Then which of the one or more of the following statement correctly describes the addition of N nucleotides during the rearrangement of the immunoglobulin heavy chain encoding gene? So what is this addition of N nucleotides? It occurs during the VDJ recombination process in the heavy chain of immunoglobulin. And this process, it is carried out by the enzyme terminal deoxynucleotidal transferase. And these added nucleotides, which are added to the VDJ junction during the recombination process, are not encoded by the template DNA. Now, this light chains, they do not undergo this N nucleotide addition because the terminal deoxynucleotidal transferase gene is turned off during the B cell development and when the light chain gene rearrangement occurs. So it is happening only in the heavy chain encoding G. So which one or more of the following statement correctly describe addition of N nucleotide during the rearrangement of the immunoglobulin heavy chain encoding gene? Addition of N nucleotide is template encoded. No, it doesn't depend on the DNA template. So A is not correct. N nucleotides are added by terminal deoxynucleotidal transferase. That's correct. The added N nucleotides are common in VD and DJ junction. Yes. Nucleotides are added by DNA polymerase II. Not DNA polymerase II. It is by terminal deoxynucleotidal transferase. Then next one. A newly identified viral protein contains one long alpha helix spanning 60 amino acid residues. The number of main chain hydrogen bonds formed in this helix is. So number of hydrogen bonds is given by the number of residues minus 4. And by this minus 4 because the first 4 residues don't form the hydrogen bonding, that is between the I and I plus 4 in alpha helix. So first 4 residues don't form this I plus 4 hydrogen bonding. So number of residues minus 4, so it will be 56. Then next one. In a lactic acid solution at pH 4.8, the concentration of lactic acid and lactate are so and so respectively. The calculated pKa of the acid. So it is given by Henderson-Hasselbalch equation, that is pKa is equal to pH minus log concentration of A by concentration of HA. Now A is concentration of lactate, it is 0.087 and HA is that of lactic acid, that is 0.01. Substitute the values and you will get it as 3.9. Then next one. If a 10 millimolar solution of a biomolecule in aqueous path length of 10 millimeter absorb 90% of the incident light at 280 nanometers, the molar extinction coefficient of the biomolecule at this wavelength is. So absorption as per Beer-Lambert's law is given by epsilon Cl. Here C is 10 millimolar they have given and length is 10 millimeter. So it is equal to 1 centimeter. Now you have to calculate absorbance, they have not given. So minus log I by I0. So minus log 1 by 0.9, 90 by 100. That 90% they have given. So that is 0.9. So minus log 0.1 is 1. So from there you have to, from this A is equal to epsilon Cl, you have to calculate epsilon is equal to A by Cl, that is 100. Then next one. Metabolic intermediates provide the backbone for the synthesis of amino acids. Okay, from Krebs cycle metabolic intermediates also amino acids are formed like in alpha ketoglutarate acid. Match the metabolic intermediates listed in column 1 with their corresponding amino acids given in column 2. So alpha ketoglutarate is the metabolic intermediate for which amino acid? Glutamate. It's given in the name itself, you can see glutarate, glutamate. So P2. Ribose 5 phosphate is histidine. 3-phosphoglycerate is serine and phosphoenol pyruvate is phenylalanine. So as per the options, it is answer A. P2, Q1, R5 and S4. Then next one. Which one of the following is correct match between the molecular properties listed in column 1 and the corresponding biochemical separation methods in column 2? So based on solubility, it is salting out, ammonium sulfate precipitation. So P3. Then ionic charge is for isoelectric focusing. Polarity is for reverse phase chromatography and molecular size is for ultracentrifugation. So P3, Q4, R1 and S2. Answer is option C. Then next one. Which of the following statements is or are correct regarding the electron motive force generated by electron transfer? It is used for the synthesis of ATP. That's correct. Chemiosmosis is the method for ATP synthesis. And this ATP synthesis, this process, it involves proton gradient or a pH gradient across the membrane. And it is composed of both electrical potential gradient and chemical gradient. So it is not used for active transport process. That is not correct. It includes a pH gradient component. Yes, that is correct. It does not include an electrical potential gradient component. It includes an electrical potential gradient component. So B and A and C are the correct options. Then next one. Which one or more of the following statement is or are correct regarding the transport and retention of proteins in different cell organelles? Mannose 6-phosphate residues are involved in targeting protein to lysosomes. That's correct. Transport of proteins into the mitochondrial compartment is aided by positively charged amino acid residues at the N terminus and internal hydrophobic segments. That is also correct. The retention of proteins in the endoplasmic lumen requires KDEL sequence at the C terminus. That's also correct. So even if the protein moves to Golgi complex, it can be brought back to the endoplasmic reticulum because of the presence of KDEL sequence. Then nuclear proteins are transported in an unfolded conformation. It is not unfolded, it is folded conformation. So D is not correct. So the answer here is A, B and C. Then which one of the following statements correctly describes fluorescence spectroscopy? The emission maxima is independent of the excitation wavelength. Yes, that's correct. The emission maxima depends on the quencher. No. Quenching affects the intensity of fluorescence, not its emission maxima. So B is not correct. The emission maxima varies with the solvent polarity. That is also correct. Then the emission maxima varies with temperature. That is also correct. So A, C, D is the answer. Then which one of the following statement is or are correct in the processing of pre-mRNA in eukaryotes? 3' 5' exonuclease activity is involved. It is not involved. It doesn't have anything to do with pre-mRNA to mRNA conversion. So A is not correct. 5' capping and addition of 3' poly A tail precedes splicing. No, it is of the order 5' capping, then splicing, then poly A tail addition. So that statement is also not correct. Splicing of pre-mRNA occurs via trans-esterification reaction. That is correct. Alternative splicing can yield different mRNA products from the same pre-mRNA. That is also correct. Then next one. Which one or more of the following statement correctly describe the changes upon addition of puromycin during eukaryotic translation? So puromycin is an antibiotic and it resembles the amino acyl end of a charged tRNA molecule and it binds to the A site and thereby cause premature chain termination. Now read the statements. Puromycin resembles amino acyl end of a charged tRNA. That's correct. Puromycin occupies the A site of the translating ribosome. That's also correct. The other statements like it occupies P site, E site, that's not correct. It occupies the A site. Then factor H, a complement regulatory protein in the plasma, binds C3b and competes with factor B to displace capital B small b from convertase. That's correct. Initiates the catabolism of C3b into inactive products. That's not correct. Then binds to C3bb convertase. That is correct. Sorry, that's not correct. Act as a cofactor for factor 1. That is correct. So the answer here is A and D. Then next one. In Michaelis-Menten equation, concentration of S is equal to 15 km. The ratio of V0 by Vmax is. So V0 is equal to Vmax concentration of S by Km plus concentration of S is the Michaelis-Menten equation. They have given concentration of S is equal to 15 km. So substitute for 15 km for V0 Vmax. I have rewritten or rearranged the equation. V0 by Vmax is equal to concentration of S by Km plus concentration of S. So in place of concentration of S, write 15 km and then you will get the answer as 0.938. Then next one. A 5250 base pair long plasmid with 10 negative supercoils would have a linking number of dash considering 10.5 base pairs per turn of bDNA. So linking number it is given by twist plus Rith. It is given that it is 10 negative supercoils. So Rith will be minus 10. Twist you have to find using the formula number of base pairs by number of base pairs per turn. So 5250 divided by 10.5 will give you 500 turns. Now calculate the linking number. 500 minus 10 is equal to 490. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.